All right, folks, thanks for joining us once again, where we're dealing with Nicholas Verhoeven once again, who does think a lot of himself, which I don't have a problem with by itself. Good. If someone backs themselves, good. However, unfortunately, if you're going to back yourself, you've got to get things right. So let's go through and see what Nicholas does, in fact, get right in this video and what he perhaps doesn't so much. Right, stick around. Here it comes.
I've seen very little content by Dr. Saladino. My
Good, keep it that way.
understanding is he's part of the carnivore community.
Then your understanding is flawed, son, right from the bat. Right off the bat, I should say. No, he's not. Paul has left the building.
And that's about all that I know.
Well, then you don't know anything.
But, as is the nature of...
Also, you'll notice there that Nicholas Verhoeven, there is underneath his name there a little PhD with a little C in brackets after it. That means you're a student, Nicholas, okay? I mean, just so we're clear on who we're listening to, this is a student, all right?
search algorithms, this video popped up in my feed, and of course I had to click on it because I just finished reading 10 scientific studies on the topic in my detailed...
Scientific studies, you say? Well, this will be interesting. Let's see how many scientific studies you actually give a competent analysis of in this video.
Analysis of LDL and heart disease. Especially considering he's a medical doctor, I'm especially intrigued.
Well, he's qualified as a medical doctor, Nicholas. I don't think he's actually practiced a single day in his life. Have you, Paul?
His very first statement. Here it is. Take it away, Dr. Saladino.
Why I don't work.
This is obviously a video from about 2 or 3 years ago, where Paul may have appeared to some to be somewhat more than that, more youthful than he perhaps may appear in the opinions of some people these days, only 3 years later.
About my quote-unquote elevated cholesterol, which should say elevated LDL, elevated low density lipoprotein. Multiple reasons. Number one, becoming more specific as I enumerate them. Number one.
Right, hear that, Nicholas? What Paul is doing is starting more general and moving towards more specific points. So this first point is kind of really just designed to allay the fears of people who have been taken in by the LDL is bad necessarily brigade, and you should get it as low as possible. There are people claiming you should get it to zero if that's possible. Of course it's not. You will die in the attempt to get it that low, but there are people saying the lower the better. And what Paul is saying here by saying, well, actually you need a certain amount to live, at minimum, there is a physiological range, we shouldn't worry, which is not really a great point, but it's more general, as Paul just said. There you go. I'm defending Salad Boy. Incredible.
One, LDL is essential for life.
Yes, good. Okay, so leave it at that.
These kids with Smith-Lemli-Opitz syndrome.
We don't need this example because it doesn't make sense to anybody, Paul. Unless our audience that's watching this video are clinicians who understand all of these syndromes very, very well, your point falls on deaf ears. It weakens your point.
Don't have enough LDL because they can't make enough cholesterol. What do for these kids? They feed them egg yolks. They give them as much exogenous, as much food cholesterol as they possibly can in hopes that that will help remedy the body's inability to make cholesterol.
Right. How does that relate to me, a normal functioning human being that doesn't have that syndrome like almost everybody on the planet, Paul? It doesn't. Okay. So you've just wasted a bunch of our time.
You're giving them tons of egg yolks. They're giving them...
You've said that.
Cholesterol.
You've said that too.
To get better. You need cholesterol to function.
You've said that.
Why would it be bad for humans if we are otherwise healthy?
There's no logic there in saying, why would it be bad for us if we need some? It completely fails to understand the basic common sense that there is a physiological range. Now, that should tell you something, shouldn't it, if you have any sense of how biology works? It means the human system has a range within which it will maintain a number of different things, including the level of cholesterol and lipoproteins in the blood. What controls this? Largely, it's the genes. The dietary intakes have some minimal impact, absolutely, but mostly it is the genes and those genes reacting to the environment in which they're placed, which sets the physiological range, which has an upper and a lower, for want of a better term, limit. So what are we worried about here? Oh, associations. Yeah. Oh, dear.
General argument here is that we need cholesterol to function.
Yeah, correct.
I completely agree.
Well done, Nicholas.
We do. I don't think anyone in the scientific community would disagree.
How would you know, Nicholas? You don't get to be a member of the scientific community until you're finished being a student and you actually have some qualifications, knowledge, and have dipped your toes into the water as an actual functioning scientist in your own right.
But that's...
Get your training wheels off first, son. Okay, that's what I'm saying.
Not an argument. The thesis is, why not worry about elevated cholesterol?
Correct.
But
we did say, or Paul did say, he was starting more general and his constructs were to be read together. So all of this is a bit pointless.
Why not worry about cholesterol? Most in the scientific community would agree that...
You've said that already, and you're still not a member of that community.
We need some cholesterol. If your cholesterol is 0, you're likely dead or on your way there.
Correct.
But that doesn't mean that we need more than we, need.
Yes, you've made your point. We agree with your point. However, we need to hear what Paul's other points are before we know whether his message is cogent as a whole, don't we?
Yes, cholesterol is part of our cellular membranes, found in the neurons of the brain, the myocytes of the muscles, the goblet cells of the intestines, and many billions of other cells. But again, that doesn't mean that once all those cells are satisfied of their cholesterol fill, that we need even more of it.
Well, what do you think the body would do in a circumstance where it needed of something? There are two options. Store it because there's any risk whatsoever that we could be in shortfall on a rainy day, or excrete it because there's no risk we could ever run short. We can always have as much as we need because we can generate all of it that we need ourselves endogenously. Which one is it going to be? Okay, if it's saturated fat we're talking about, well, if we have absolutely more than you need, your body will still store it because there is a risk that we might run short on that on a rainy day. So that's what the body does. With cholesterol, which the body is perfectly capable of manufacturing itself from the ground up, basically, from acetyl coenzyme A units, no need to store more than we need. And in fact, there is a transporter on or in the alimentary canal which can direct excess to the bowel for excretion. Exactly what you'd expect. So how much cholesterol or how much lipoprotein or any fraction of the lipoproteins that your body needs right now is under the control of the genes. Those genes set what those levels are on the basis of what is in fact needed. Need is determined by the body's need. Actually, at the time, not some standard imposed by the so-called scientific community who have decided what so-called ideal levels are. And given that they have not supplied a scrap of evidence for causality, and I know you're going to talk about Mendelian randomization in a minute, this will be hilarious, boys and girls, the argument is not made here, is what I'm saying, by those who say LDL is necessarily problematic at the high end, because there is no indication of that. Right, what's next?
Which is the issue. It's like saying, we need water, so therefore I don't worry about my excess water intake. Okay, but if you consume...
Yes, we understand the concept, Nicholas. Unlike many people, and I suppose that includes many of your listeners, we ain't stupid. We get it.
30 liters of water, then you die. No one would say water isn't vital to life, but having none is also lethal, just as having too much is sure to have you visit...
Yes, we get it.
...your gravesite as more than just a tourist. So, I agree that we need cholesterol, but that isn't an argument.
You've said that.
For having excess cholesterol.
You've said that too. We understood the first time you said it.
Second thing, LDL has critical immune functions in the human body, which I'll show you guys a little later.
Okay, fine. Interesting. There are many roles that cholesterol plays. Immune modulation is in fact one of them, yes, but there are a milieu of roles that cholesterol plays precisely because it is so ubiquitous.
It interrupts quorum sensing, which is the communication between bacteria. And we know in both animal and human experiments that if you decrease the LDL, animals get more sick. And in humans who have lower levels of LDL, they have increased rates of communicable infectious disease.
Okay, great. An association. It's interesting. But so what?
So LDL is an immune particle. These are two critical features of LDL. Why would something that is essential for human life...
You've said that, Paul, already, remember?
...be bad for us if we are...
You've said that?
...otherwise healthy?
LDL has immune functions. I think this is a cool point. When we consider low-density lipoproteins, we consider the trafficking of cholesterol molecules and triglycerides across the body. The lipoprotein is the vessel, and the core is the inside of the package, containing all the ingredients our cells need, dropping off various molecules for their cellular workings. However, it always fills me with a bit of childlike excitement when...
Well, it's not that many years since your childhood, son, so, it's not surprising that you could maintain some of that.
We look at non-canonical mechanisms of our bodily processes, like LDL as an immune modulator. I did a bit of digging into the mechanism and found a few studies on the topic.
You mean some theoretical matter on the topic?
So, quorum sensing is simply bacteria signaling to one another their population density. They will release molecules that bind surrounding bacteria and either activate or inhibit their growth. What's ridiculously cool is that LDL particles are associated with infection. According to this study, the researchers point out that people in a clinical setting experience worse infections if they have low LDL levels.
Sure.
Which speaks exactly to Dr. Saladino's point. The reason is that the ApoB
You mean the theoretical reason is, the hypothesized mechanism is...
molecule attached to LDL particles can bind these molecules released by the bacteria and inhibit their ability to bind other bacteria, thereby reducing their signaling. Of course, if you have no LDL, then you have much lower ApoB, and the bacterial population can grow uninhibited, at least by this insular mechanism.
Right, exactly. So we're talking about a milieu of different interactions, of different interworkings, of different parts of the metabolic pathway, etc. So looking at this one thing in isolation, conducted almost certainly in a test tube away from the workings of a free-living human body, so what? This isn't science. This is speculative. Well, it's called mechanistic speculation is what this is.
Essentially, LDL particles sequester endotoxins released by bacteria. Isn't that really super freaking cool?
Only if you have childlike enthusiasm.
So, Dr. Saladino is right. LDL aids in the process, and low LDL is associated with worse health outcomes.
Right, but associations don't prove anything.
However, it should be noted that some people have low LDL in the hospital because other diseases drive LDL levels down.
Oh, yes, reverse causation, you say. Goodness gracious me.
Not because they actively try to keep their LDL at such low levels and thereby incur this added infection risk. Anyway, I guess...
No. There are no studies that inform us on risk as that relates to outcomes of a hard health nature in human beings over any period of time under any sort of control and observation. Risk is a cause and effect statement. I'll come back to that in a bit. If you want to make a cause and effect claim, risk, you must supply cause and effect evidence for that. And that means proper scientific experimental work conducted under the disciplines of science. And that's not what we're looking at in the literature, Nicholas.
We can move on. Oh, wait. What's this? Another point being made by the researchers of the exact same study. Wait a minute. This can't be right.
Oh, get on with it, Nicholas. We don't need this theatre. None of us are amused.
Check this out. Together with previous reports demonstrating that hyperlipidemia also impairs lung innate defense, these results suggest that maintenance of normal...
Right, so with regard to this one isolated relationship, there seems to be potentially an optimal level physiologically for LDL. It's almost like we have a set of genes that basically encode a bunch of instructions which can be moderated upward and downward in terms of how active the process is, that all function together in such a way as to do the best job they possibly can to maintain the homeostasis of the organism at large, including the level of LDL that is needed in the blood at that time. And it goes about its merry business of doing that. An association with potentially increased incidence of some disease process of the lungs with LDL levels in some way is not an experimental study proving that this effect is occurring in free-living humans. I should say whole humans, not free-living, because you'd need to put them in a laboratory lock-in situation to do this study, wouldn't you? That's why it hasn't been done. because it's not ethical, practically possible, or financially possible. There is no cause and effect data extant. This is a theory. The hyperlipidemia / cardiovascular or any other kind of disease process theory.
Normal serum lipoprotein levels is necessary for optimal host innate defense in the lung. The researchers are saying that while, yes, very low LDL
we get it,
is linked to worse bacterial infections, having elevated LDL is also linked to worse infections through impairing the immune system. Then they go on to say that maintaining LDL in the normal range is the
normal range? No. There will be an individually decided optimal level at that time for that individual. Normative values have nothing to do with an individual person's prospects in any way, shape, or form. Nicholas.
Sweet spot in relation to these infection outcomes. Well, it's almost as if having too little or too much can be detrimental.
That's true of anything, though, including oxygen, water, sunlight. This is a vacuous point.
Where have we heard that before?
The body can excrete excess cholesterol and excess lipoprotein breakdown. What is your problem, Nicholas? Other than you imagine for some reason that human physiology would be so deranged as to not be able to cope with changes in circumstance, the environment in which you place your genes at any given time, and to then behave in the way best conduced to keep you both alive and as healthy as possible while doing so.
It's like saying, we need water, so therefore I don't worry about my excess water intake. Okay, but if you consume 30 liters of water...
the repetition. It's incredible, isn't it, kids?
Then you die. No one would say water isn't vital to life, but having none is also lethal, just as having too much is sure to have you visit your gravesite as more than just a tourist. So, I agree that we need cholesterol, but that isn't an argument for having excess cholesterol.
Yeah, but you haven't provided an argument for not having so-called excess, Nicholas. That's what you would need to be able to do.
Third thing is perhaps the most important. Context is everything. Context is everything.
You just said that, Paul.
LDL, does it directly injure the endothelium and cause atherosclerosis, or is it a bystander, or is it a repair mechanism that is getting pulled in?
There's many possibilities there.
Because if it is either of the latter two cases, it could associate with cardiovascular disease in those people who are metabolically unwell, but not be causing it. And I believe that
correct.
That is the oversight that is being made by physicians in the medical community over and over and over. If you repeat a lie often enough, it becomes the truth. I've even had preeminent lipidologists debate me on Twitter, which I don't really spend much time on anymore, saying everyone knows LDL is harmful.
No, they don't. A lot of people think they know that. They are in error.
It's just, 99.7% of the world's lipidologists believe LDL is harmful. Who are you to say that it's not? Well, I'm a human being that is thinking about this properly. Well,
Paul, you might be thinking about this particular issue properly, but you don't do yourself any favors by thinking so wrongly, so erroneously on other topics that reduces your credibility beyond repair.
Not to become dogmatic, which is to sacrifice curiosity for the sake of consistency.
No, it isn't. That's your opinion. If you are correct, absolutely unassailably correct about a given topic, then you are within your rights to plant that flag and say, I am unassailably correct here. Welcome all comers. Come and try and take this flag down. You will fail. That's not dogmatic. That's being prepared to accept what is objectively true until proof is provided that it is not objectively true.
I, too, am a human being that is thinking. I didn't learn about LDL being harmful to our blood vessels merely by being taught that in my master's and PhD.
You don't have a PhD, son.
But rather because I read a multitude of studies on the topic.
Oh, right. You read some studies on the topic, and now you're an expert. Okay. Let's hear about these studies that you've read.
My analysis is public, albeit long.
And erroneous.
Anyway, let me address this first aspect of point 3, and then I believe Dr. Saladino makes another point that I'd like to address. He mentions that LDL is not a causative agent, to cardiovascular disease.
Not as popular, probably too strong. The correct scientific position, the unassailable position to take is there is no evidence that LDL is causative in any negative health outcome of any kind, i.e. elevated lipoproteins. That is a fact. There are no evidences available which can establish the case. There are a number of weak, poor inferences available for someone who is either theologically already determined to take a given position, or they are an unconscious incompetent with Dunning-Kruger syndrome who actually doesn't understand what is and what is not science. Let's see which it is.
Blood vessel atherogenesis. I'm not going to pull up every study that I analyzed, but I will point out one that had a great impact on me.
Right, so you've been impressed by Mendelian randomization. Ergo, that's all we need to know about you, Nicholas. You are a fool. You have mistakenly assessed yet another form of associative mishmash without control as science in some way, purely because it looks at genes, which is oh so technical and therefore must be good science.
It's called a Mendelian randomization.
Right, so we're done, Nicholas. Your credibility is now destroyed. If I hadn't already been by the multiple videos I've made about your ridiculous nonsense hitherto.
Wow. Essentially, the researchers of this study took people with a gene mutation.
No, not a gene mutation at all, Nicholas. A whole bunch of different gene mutations lumped together into this one bucket of people called low LDL SNP people, or whatever they call them in these studies. There are all sorts of different labels that are appended to these hugely genetically disparate group of people, actually, who have different SNPs that lead, among other things, universally, to expressing a lower level of LDL than the next person who doesn't have any of these mutations. Right. That's all. No control over these individuals. No control over the covariates, confounders, collinearities, all the things that destroy this study's ability to do anything except express an association of the incidence of any number of different factors with the fact that they have been put arbitrarily in this one bucket of people, even though they're quite genetically disparate. This is not good science.
In their genome that caused them to have lifelong reduced LDL cholesterol, and compared them against a massive other set of individuals that do not have this mutation.
Great. And were the two groups homogeneous in terms of their expression of other things that we know might affect that person's individual likelihood of developing heart disease, atherosclerosis in their lifetime, or any given period of their lifetime? For example, were the people with the SNPs of a similar stature, body composition, prevalence of diabetes, and any number of other factors? The answer is no. We do not have two otherwise identical groups of individuals. There are confounds, covariates, collinearities. The outcome of a human being in terms of their health is a rich, multi-thread, tangled, meshy thing that is inextricable in terms of causality unless you take some genetically identical individuals and you split them into two groups, being one twin and the other twin, multiple numbers of twins, and you lock each group in separate laboratories where everything absolutely and utterly is controlled except for the one thing that you want to point the finger at, that being the LDL exposure over the lifetime. If you do not do that, you have not established cause and effect. Those are the rules. I'm sorry, kids. I don't make the rules. I happen to agree with the rules because the rules require you as a scientist to exercise discipline and to only state causality where it is established. And with regard to low-density lipoprotein or any sub-fraction of that, there is no established cause and effect case, nor even any decent inference. We're done with Mendelian randomization. It is not good high-quality science. It is rubbish.
Thereby having normal LDL level. So, irrespective of habit.
Further, before we move off that one also, often in the low LDL-C group people, we are expected to believe that because they carry that SNP, then their individual LDL-C is indeed reduced. How many of the people in these groups have they traced the lifelong exposure to LDL, i.e. by taking a blood test off that individual every single day of their lives, shall we say? None? At all? Okay, good. I hope we're getting closer to the realization that little boys who've played in the sandpit a little bit should not therefore think themselves to be king of the castle in terms of the arbiter of the science, because it's clear that little boys with such little spades and little buckets should actually probably concentrate on what they're doing, learning how to use the spade and the bucket effectively, not claiming to be king of the castle.
Nutrition, dress, alien abduction, smoking, dancing ability, and every other factor that you can think of
has been completely ignored. Ergo, this is not a scientific study that can inform us on cause and effect. Wow.
The people with the gene mutation have lower LDL.
No, they don't. They're imagined to. Again, rewind a few minutes and listen to me talking about or asking the question as to how many of them have been traced to prove that that is so.
And across thousands of participants with these gene mutations, they had huge reductions in heart disease risk.
False. Number one, the reductions in incidence were not huge. They were minuscule. And this data is associative and therefore cannot inform on risk. Why am I saying that? Why am I saying risk is a cause and effect statement? Well, that's very easy. Let's go here. If you say to me that my risk of anything is affected by the concentration of anything in my blood, then you better be prepared to go to guns and back that up with some experimental evidence that it's so. Just because an association, piss weak at best, has been, well, not even observed because they adjust before they report the risk, which, in other words, they fabricate data completely, change the results 180 degrees out of phase with what was actually observed. That's fine, apparently. Apparently that's good science, according to Mr. Verhoeven here. No.
Risk, relative and absolute risk.
No, this work cannot inform on risk, period. Not one jot.
We can see that evidenced here.
No, that's not evidence. That is an associative plot with a trend line drawn through it and some indication of the error around that relationship. What's not on this chart is any information whatever concerning cause and effect. Nothing at all. We're done with this. The plot could just as easily be a plot of the incidence of sunburn occurring at any given beach resort, any given day. On one axis and on the other axis, you might find the sale of ice creams at the same beach resort. So do we need to outlaw the ice cream? Is that what we need to do here? I mean, you started reasonably, Nicholas. You pointed out that Paul's argument was pretty fragile, as it is, and that's no surprise. And then you yourself go completely off the rails and get excited about Mendelian randomization, which is just another form of association which cannot inform on causality, not one jot. Sandpit, bucket, you know. Right, what's next?
With the boxes being samples of hundreds or even thousands of individuals with these gene mutations. Called SNPs or SNPs.
So?
The horizontal axis is the...
But don't forget, we also have no idea about these people's actual expressed LDL levels throughout their lifespan. So this is an association of a thing that they're calling risk, which isn't, because it can't inform on risk. This is an incidence report. Is it adjusted in any way? Is it fabricated? Probably. And on the other axis, we've got lower LDL-C, which is something that they haven't measured. So this is just a fantasy, Nicholas. If you want to take a one-off reading of cholesterol taken at the beginning of a follow-up period in a study as indication of an LDL-C level across a study period of multiple years, I want some of whatever you're smoking. If you think that's remotely valid and that this is anything other than a fantasy, Nicholas. You've been taken in. You've been fooled precisely because your level of sophistication in science is none.
Reduction in lifelong LDL.
We don't know that.
And the vertical axis is the risk reduction in
No, it isn't.
Heart disease. No matter the version of the mutation they had, they all had lower risk
No, they didn't, because this chart cannot inform on risk.
due to lower LDL.
You said due to. That's a cause and effect statement directly, Nicholas. No.
Before you jump down my throat and tell me that I don't know association from my own rear end.
Well, too late. I've already done so because it's clear that you don't. It's also clear that you wouldn't know your own elbow from your rear end if I labelled both of those and taught you how to read, son. Pull your head in. Finish your studies. Concentrate on what you're doing, for goodness sake.
I would encourage you to learn a bit more about Mendelian randomization.
I have done. I think I've been quite clear in this video and several other videos as well on the topic. This is rubbish, Nicholas, and you're a fool.
Trials. They're incredible studies.
No, they're not. They're incredibly farcical.
That can, if done correctly,
No, they can't.
virtually bridge the gap between association and causation.
There is no bridge, virtual or otherwise. The only way to that land of causality is through a bridge that is already there, a land bridge. The rules, actual science, experimental works conducted under control. Nicholas, no associative study can inform on risk. Period. Those are the rules. That is the scientific discipline. And until you get that through your head, you would fail any assessment or defense of your PhD, which isn't finished yet. If I'm your assessor, and you better hope I'm not, because I still do assess PhDs, no, son, you failed to understand the first lesson that you should have learned in the first half hour of your first undergraduate degree. Association cannot inform on causality, period. Let's go.
Okay, I have other studies in mind, but let's leave it there and hear what Dr. Saladino says next.
Hopefully you guys are all still with me at this point. And the
No, Paul, you left the building several years ago, son.
thesis that I will advance in the remainder of this podcast is that I will try to prove to you that LDL does not directly injure the endothelium of the blood vessels and that context is everything.
Well, it's clear that it doesn't directly cause injury. He's going to give the high pressure, low pressure, one side of the vasculature versus the other side of the vasculature argument, which is valid. The fact that it's devastating to the posit of this ridiculous boy here is neither here nor there. Neither is it here or there that a lot of so-called preeminent so-called lipidologists agree with what Nicholas is saying. That doesn't make it right. The facts are unchanged by any given person's interpretation of those facts.
For it. Settle in.
The metabolic dysfunction, also known as insulin resistance, is the proximate
Is a construct, is an idea put together by some people trying to conceptually understand what is a complex interaction of various metabolic functions. And it erroneously claims that this thing, insulin resistance, is problematic, is pathogenic in some way. No. It's covered in great detail on my channel already if people want to go and have a look.
event, the first event of atherosclerosis. And if you avoid that,
No. So-called insulin resistance, the construct, is not the first domino in the chain of events leading to atherosclerosis heart disease. Not at all.
if you are metabolically healthy, you can have a ton of LDL in your body and not injure your endothelium.
Unless, let's say, you're hypertensive, in which case you can't. Paul. So, false.
You actually may benefit from that from cell membranes, from a hormonal perspective, and from an immunologic perspective.
Sure, that's possible.
We've covered this already. Even if you're metabolic
Oh, right. So now you want to complain about people repeating themselves, Nicholas, do you?
healthy, LDL is an independent risk factor. Additionally, while I agree that
LDL is not an independent risk factor because the word risk requires you to supply me with some experimental evidence. Show me a study, Nicholas, where a statistically powerful group, a large group of genetically identical individuals are separated at birth or beforehand. Good luck with that. Kept in laboratories under strict control in every aspect of their lives for their entire lifespans, except for this one thing, exposure to the LDL, shall we say, until you can show me a study of that nature that would establish cause and effect. You have not established cause and effect. Those are still the rules.
is critical for cell membranes and seems to be a benefit to immunity. We discussed how too much is still not needed and can be counter to some of those points.
So, at a very, very high level, let's ask this question. If LDL is directly injurious to the endothelium, and I believe it must be directly injurious to the endothelium in order to cause atherosclerosis de novo from the beginning, from scratch. Why do we get atherosclerosis only in arteries and not in veins when the veins are within a physiologic system of low pressure?
Bam. That's it. End of LDL hypothesis as a causal agent. Done. The most sensible thing I've heard Paul Saladino say in years.
Why do we typically experience atherosclerosis, the buildup of plaque in our arteries and not our veins?
Because mechanical, chemical, and immune-based damage and or inflammation to the vascular epithelial cells is required for any atherosclerosis development to occur. Those things don't occur in the veins precisely because one of those vital ingredients is removed entirely by the fact that the low pressure side of the vasculature is A, low pressure, and B, not pulsatile. So it's like the fire analogy where you say, well, fire needs fuel, oxygen, and heat. Remove any one of those three and the fire dies. It's the same with the development of atherosclerosis, the disease atherosclerosis. Knock out any one of those absolutely required causative agents, it will not happen, irrespective of your level of LDL-C. Ergo, LDL-C is not the cause. Period. We're done. Finish. End of discussion. This is very simple.
Well, he brings up a good point.
Unassailable.
But before he does, I should mention that arteries and veins are also structurally not the same.
So what?
Arteries have far more smooth muscles lining the artery structure, and smooth muscles are implicated in the progression of atherosclerosis.
Among other things, yes.
So there is one potential reason. The second reason is a good one by Dr. Sound.
Okay, then, if that were the case, Nicholas, my foolish little bucket-wielding spade maniac, atherosclerotic lesions would develop evenly. There would be a homogeneous level of atherosclerotic lesioning in the high-pressure side of the vasculature. Wouldn't there? Is that what we see? No, it isn't. Go and look up where atherosclerosis develops, Nicholas. All right? You'll learn a lot.
Dino, let's hear him out.
If LDL were injurious to the endothelium, which is the same in veins and arteries, why does it not cause atherosclerosis in veins? It will cause atherosclerosis in a vein if you put that vein into an arterial circulation.
Bingo. So it looks like the pressure being exerted, thus the mechanical damage being done to those epithelial cells in those particular areas where atherosclerotic plaques do develop, i.e. the areas of turbulence within that system, where the transit time of blood past those areas is thus slowed by the turbulence. So the elongated transit time combined with the elevated pressure, bingo, a vein without that smooth muscle imposed into the arterial side of the system, bingo, atherosclerotic development. Goodness, I think this is pretty clear.
What we know is that the higher pressure of our arteries, 120 millimeters of mercury during systole, which is when the heart...
That's so-called ideal, though, Paul. Most people have elevated levels above that, especially on a standard diet. Very dangerous over a lifetime.
...contracts 80 millimeters of mercury during diastole versus much lower pressures in the veins does damage the endothelium. Aha. So what we need for atherosclerosis to begin is a damaged endothelium. And I think...
Right. Not LDL-C as a causative agent. No. Damage, inflammation, immune response, chemical derangement of various things, including LDL. We'll get to that if we get to it, though.
That is the key here.
He mentions that arteries are in a high-pressure situation. And that's true because they're taking the greatest pressure.
Oh, God. Seriously, son?
Pressure load from the heart pumping, because they are the circulatory system nearest the highest pressure moment of the heart pumping. I think this is a great point, and no doubt the pressure difference could easily contribute to the issue in arteries as it does not in veins.
It's not could be. It's obvious. It's a slam dunk, you foolish boy.
The LDL isn't damaging the endothelium, or we would see de novo atherosclerosis in veins.
Bam. Drop the mic. Correct.
The endothelium must be damaged for LDL to become involved in the atherosclerotic process.
Correct. But how is it involved?
Right?
Yes, correct.
Simple. That is just something that I want you to ponder over. that you must have damaged or dysfunctional endothelium. And I think the endothelium in our arteries is always being damaged by the high pressure.
Especially in some people.
Again, a very logical point, and there's likely some truth here. However, it still ignores multiple other factors, like the structural differences between the veins and the arteries.
I've dealt with that one for you, Nicholas. Why then does the atherosclerosis not develop evenly in the arteries? Why does that not happen, Nicholas? Explain that, if what you're saying has any validity, which, surprise, surprise, son, it doesn't.
Or the LDL load in one circulatory system, arteries, versus the LDL load in the other, veins. Or,
well, do you have any data on that, son? No?
Or the proteoglycan levels, which are proteins that attract the LDL particles through the endothelium, the first layer of cells of the blood vessel. Either way, blood pressure is no doubt a factor.
Yes.
And I would say this. In fact, I would say this very strongly.
Ooh.
Atherosclerosis does not exist without insulin resistance.
Well, that's a foolish thing to say, Paul, and it's false. Notwithstanding the arguments around whether or not insulin resistance actually exists or is conceptualized even remotely correctly by the vast majority of people that talk about this, no, there is evidence that people develop heart disease without any apparent so-called insulin resistance. The one I'm talking about with Paul Saladino says something perfectly unassailable and then backs that up immediately with something ridiculous, something foolish, incredible.
No one ever had a heart attack. Not a single individual.
Ridiculous, Paul.
Well, that's not true. I can't say that. But
there you go. No, son.
The vast majority of people who have heart attacks, this is atherosclerosis-induced plaque ruptured heart attacks.
Yes.
Atheroscleromas are insulin resistant, whether they know it or not.
Perhaps, depending on how you conceptualize this so-called insulin resistance and how it's so-called measured, given that you can't measure it, it's a construct, you have to construct it from other measurements. Give me the name of a company that has created an insulin resistance meter that we can do a finger prick test and get a beep, 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 boop, 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 boop. There's your insulin resistance number. Because insulin resistance is not a substance. It's not something you can measure. It's a construct, an idea.
This is pretty easy to test. I do agree that people who are insulin resistant are likely people with elevated LDL and likely have other dysfunctional components of their physiology.
So what you're talking about there is confounding covariation in some way, which is the very foundation of the reasoning behind what we're saying here, which is that association cannot inform on causality.
That can contribute to the rapidity of atherogenesis, cardiovascular disease. However, heart disease can form independent of insulin resistance.
Right, so there you go. Paul Saladino tripped up by a student.
One of the easiest ways to test this is to return to genetics. Instead of looking at people that have favorable mutations like we did in the Mendelian trials, we can look at people...
No, you didn't do that. You just bunched a whole bunch of... Well, not you, because you've never done a Mendelian study, actually, Nicholas, because you're a student. What was done by people doing those Mendelian studies was bunch of people together who happen to have a similar SNP, assume that their LDL is affected in the way that would be predicted without testing it, and then look at their incidence of heart disease and report that once we've adjusted it to something different. Good science. What's next?
People who have an unfavorable gene mutation. In this case, we're looking at people who are characterized as hypercholesterolemic.
Same arguments as what we've dealt with earlier regarding the low expressors. We have covariates, confounders, collinearities here. This is not an experimentally controlled study.
Which means they have a genetic disposition for increased LDL particles.
Was it measured on a daily basis to establish the facts and thus establish the proof beyond all reasonable doubt? No.
And cholesterol levels. So let's address this really quickly. A meta-analysis of 18 studies investigated the risk of heart disease.
No, they didn't. There are no studies that inform on risk. Because if you say this study informs on my risk of anything, you're mad. Number one, what were the age of the participants in such of these kind of studies at the outset? Often as high as 60s, 60 plus years of age. So we've got 60 years of confounding. Things that will affect someone's heart health outcomes in their 60s, 70s, and 80s, should they live that long or longer. We don't have a study that's able to inform on cause and effect here. People live all sorts of lifestyles, irrespective of what their genes are, and genes react to the environment in which they're placed. Ergo, we're confounded here. We're done. Still.
In those individuals and found that in a quantification of risk.
No, these studies cannot inform on risk, and I will correct it every single time one of these buffoons falsely claims that a study exists that can inform us on risk, because there isn't one.
Every study in the cohort showed an association with increased risk.
No. Number one, the associations reported were adjusted, i.e., an association of someone's fantasy with something else, which they're calling risk. No.
Except one, as seen here. Now, in full transparency, this type of analysis is not as ironclad as the Mendelian trial.
The Mendelian trial is not ironclad. The only thing that's ironclad here, Nicholas, is your status as incompetent to comment on it.
So there is a potential for confounding factors.
Hello!
This is exactly Dr. Saladino's point.
Just like there is in the Mendelian studies.
Wait, his point is that the confounding variable in all of these analyses is insulin resistance.
Which is a construct. They go completely nonsensical.
Diabetes.
It's diabetes and insulin resistance, not the same thing. Diabetes is a clinically measurable pathology by means of measuring an actual physical substance and reporting its concentration as being X, Y, or Z. See how that's much more valid than making something up like, insulin resistance.
As you heard, without underlying insulin resistance, there is no risk of LDL being a causative factor to heart disease.
It's nonsense. The whole sentence is nonsense.
Well, I have some awful news. My bedside manner isn't quite as polished as a clinician, so you'll have to excuse
well, why would it be? You're a student.
me if I rip the Band-Aid right off and offer the diagnosis. There have been studies that have looked at the relationship between individuals with hypercholesterolemia.
How has that been established that a person has hypercholesterolemia, Nicholas? With a one-off test one morning?
And diabetes. Much to my
How is diabetes defined and diagnosed, Nicholas? Exactly.
own surprise, there's actually an inverse relationship, meaning
Of what strength, reliability, and so what?
people with this genetic condition tend to have lower rates of diabetes. There may be
Can you back that up with some actual data? Because you haven't provided any here. You've given us two little cutouts of the title of two papers that we'd have to go and read to see whether or not they do that.
a genetic overlap that causes this inverse relationship. So
Right, so genes interact not only with the environment they're placed, but with each other, given the fact that genes affect the very environment in which they themselves sit.
to summarize, genetic disease that increases LDL increases risk of heart disease.
No, that's a statement of cause and effect that cannot be supported.
And that genetic disease reduces diabetes risk, the exact opposite of what
No, because there are no studies that can inform on risk. Still, so false again.
Dr. Saladino mentions. But I'd like to hit this argument with one more chair, if you'll allow a wrestling analogy.
We will not allow it.
If we look at this same population, people with genetically elevated LDL levels,
Again, how has that been measured, tested, and verified as being the actual circumstance in play? Just by people having a snip? No.
and we go way into childhood, we see cardiovascular disease in children. The odds that they've
Yes, because of damage and inflammation to vascular epithelial beds, firstly, followed up by an immune response to chemically deranged, i.e. non-native lipoproteins and other factors.
also develop diabetes by the ripe old age of 5 or 8 or 10 is quite small. And even if you can find some that have, they all have astronomically high LDL levels.
Astronomically high is a value judgment statement and has no relevance to a scientific discussion. It's just your opinion, Nicholas.
So, which factor is more prevalent? Look, I'm not saying that diabetes and insulin resistance as a whole do not contribute to heart disease.
Of course, well, diabetes absolutely does. Insulin resistance is actually at play precisely in order to try and prevent that, among other things. It's actually adaptive and not a pathology.
My only argument, and it bears out massively in the data, is that LDL is one independent risk factor of...
No, it isn't. Just because that's been stated by so-called researchers who invariably are paid a lot of money to say that, it is not actually something that has been established by science. Risk is a cause and effect statement. If you want to say my individual risk of anything is related to any factor, you are implying a cause and effect relationship. So show me the experiment that backs it up. No? Okay, good. Are we done here?
Heart disease, even if it isn't the only one. Overall, I disagree with Dr. Saladino.
So do I on many issues, but not on this one particularly.
Except on a few points, like the blood pressure issue impacting arteries more than veins. If you'd like to see me do more of this kind of science fact-checking...
No, what I'd like you to see you do, Nicholas, is I'd like to not see you do anything at all until well after you've completed your PhD and your postdoc, if you're going to do one of those, or until you've been teaching to both undergraduate and postgraduate students and yourself researching as an independent scientist for probably at least 5 years, frankly. Until then, I'd like to see you remain absolutely silent on the YouTubes about science. And I'd like to also see you stop representing yourself as PhD, just putting a C after it, which stands for candidate. That means you're a student. Don't claim to have credentials that you don't have.
Check out more of my videos in my Spotlight series.
We won't be doing that.
And if you're completely uninterested, check out some of my other content.
We won't be doing that either, except to correct it where it's wrong.
If you're still uninterested, maybe I'll do a little dance in the next video.
Oh, God, please don't.
To keep you entertained.
No, that won't work.
Dance with you in the next one. See ya.
Right, there you go. What a load of absolute fetid dingo's kidneys. What a load of rubbish from a boy who ought to be doing his studies, frankly. And then when he's finished with his studies for the day, perhaps he ought to help his mummy with the dishes. Until then, I will see you next time where someone else will be wrong on interwebs because it doesn't look like slowing down anytime soon, does it? No. Right, see you then. Ciao.